Modern humans, known scientifically as Homo sapiens, represent the culmination of millions of years of evolution. Our species did not simply appear, but rather evolved from a succession of earlier hominin forms. This evolutionary journey unfolded over vast periods, characterized by significant biological transformations. Our lineage diverged from the one leading to modern chimpanzees, our closest living relatives. This pivotal split occurred roughly 4 to 8 million years ago in Africa. Genetic studies and fossil evidence provide robust support for this timeframe, marking a crucial branching point in primate evolution. It is important to understand that the ancestral ape at this divergence point was neither a chimpanzee nor a human in its modern form. Instead, it was an earlier species of African ape, possessing a mosaic of traits that would eventually differentiate into two distinct evolutionary paths. This common ancestor shared characteristics with both lineages, predating the specialized features we observe today. This foundational split established the trajectory for what would become the human lineage. From this point forward, unique adaptations began to emerge, distinguishing early hominins from other primate groups. Our understanding of this period is continuously refined through ongoing fossil discoveries and advanced genetic analysis. Over these millions of years, several key evolutionary milestones defined the hominin journey. One of the most significant was the emergence of bipedal walking, the ability to walk upright on two legs. This adaptation freed the hands and offered advantages for savanna dwelling. Alongside bipedalism, there was a notable development in brain size and complexity. Over time, hominin brains expanded, leading to enhanced cognitive abilities. This increase in cranial capacity facilitated more sophisticated thought processes and problem-solving skills. The development of complex tool use also marked a critical evolutionary step. Early hominins began to craft and utilize tools for various purposes, from butchering animals to processing plants. This innovation demonstrates increasing cognitive capabilities and a growing mastery over their environment. Furthermore, symbolic communication gradually evolved, eventually leading to complex language. This ability to convey abstract thoughts and shared knowledge transforms social structures and cooperative behaviors. Symbolic thought underlies much of human culture and innovation. These profound traits, including bipedalism, larger brains, complex tool use, and symbolic communication, did not appear suddenly. Instead, they emerged gradually, evolving across numerous hominin species over millions of years, shaping the trajectory that led to Homo sapiens. Our genetic lineage provides powerful insights into human ancestry. The concept of mitochondrial Eve refers to the most recent common maternal ancestor from whom all living humans descend through an unbroken line on their mother's side. Our mitochondrial DNA was passed down through generations. Similarly, Y chromosomal Adam denotes the most recent common paternal ancestor for all living men. His Y chromosome has been passed down directly through an unbroken line from father to son. Both individuals lived in Africa, though at different times. It is crucial to emphasize that neither mitochondrial Eve nor Y chromosomal Adam were the sole humans alive during their respective eras. They were part of larger populations, but their specific maternal and paternal lines happened to survive unbroken to the present day. Other lineages simply died out at some point. Their significance lies in the unbroken survival of their specific genetic markers. These genetic markers, mitochondrial DNA and the Y chromosome, provide powerful tools for tracing human migration patterns and population histories across the globe. Comparative genome research provides profound insights into the genetic basis of differences between humans and other primates. Studies comparing human and chimpanzee genomes reveal a remarkable degree of similarity, approximately 98 to 99% identical. This close genetic relationship underscores our shared evolutionary history. However, the differences we observe between species stem primarily from changes in gene regulation. These are alterations in when and where genes are expressed, rather than the emergence of entirely new genes. A small number of regulatory changes can have profound effects on development and morphology. Instead of acquiring entirely novel genes, subtle modifications to existing genetic instructions drive evolutionary divergence. These regulatory shifts dictate how shared genes are utilized, leading to distinct anatomical, physiological, and cognitive traits. Understanding these regulatory changes is key to deciphering human uniqueness. The remarkable diversity observed within the human species is shaped by several powerful evolutionary mechanisms. Adaptation to local environments plays a critical role as populations develop traits that enhance survival and reproduction in specific ecological settings. This process is driven by natural selection. Genetic drift also contributes significantly to human variation, particularly in isolated groups. This random fluctuation of gene frequencies can lead to certain traits becoming more or less common purely by chance, especially in smaller populations. Migration patterns further mix populations over time, introducing new genetic variants and creating clinal distributions of traits. One prominent example of adaptation is the variation in skin pigmentation. Darker skin is an adaptation found in populations living in high UV regions, such as equatorial Africa. Melanin provides protection against harmful ultraviolet radiation, reducing the risk of skin cancer and folate degradation. Conversely, lighter skin tones are prevalent in regions with lower UV exposure, like Northern Europe. This adaptation facilitates vitamin D synthesis in environments with less intense sunlight. These variations demonstrate a clear link between environmental pressures and phenotypic expression. Another compelling adaptation is lactose tolerance in adulthood. Historically, most humans lost the ability to digest lactose after infancy. However, in cultures with a long history of dairy farming, like certain European and African populations, a genetic mutation emerged that allows for continued lactose digestion. This provided a significant nutritional advantage. High-altitude adaptations offer further evidence of natural selection at work. Populations residing in mountainous regions, such as Tibetans and Andeans, have evolved unique physiological traits to cope with lower oxygen levels. These include differences in lung capacity, blood oxygen saturation, and metabolic efficiency. Tibetans, for example, exhibit genetic variations that lead to higher oxygen saturation in their blood, even at extreme altitudes. Andeans show different adaptations, including larger lung volumes and higher hemoglobin concentrations. These represent distinct evolutionary paths to address similar environmental challenges. Geographic isolation has profoundly impacted human genetic and phenotypic diversity. Secluded populations cut off from significant gene flow often develop unique features that distinguish them from other groups. This isolation allows for the accumulation of distinct genetic profiles through drift and localized adaptation. Examples include the Andaman Islanders, who exhibit unique physical characteristics due to their long isolation in the Indian Ocean. Similarly, the Highlanders of Papua New Guinea and various Himalayan groups show genetic distinctions reflective of their geographic separation. These populations offer living laboratories for studying the effects of isolation. Amazonian tribes, often living in remote, untouched rainforests, also display remarkable genetic distinctiveness. Reduced gene flow, meaning limited interbreeding with outside populations, is the primary mechanism through which these traits become more pronounced and distinct. This lack of mixing preserves unique genetic patterns. In stark contrast, genetically mixed regions are characterized by long histories of extensive population interaction. Areas with prolonged periods of trade, migration, and the establishment of empires typically exhibit the most diverse genetic landscapes. These regions are melting pots of human ancestry. The Mediterranean basin is a prime example, having served as a crossroads for countless civilizations and trade routes over millennia. Its populations reflect a complex tapestry of genetic contributions from Europe, Africa, and the Middle East, demonstrating repeated blending of traits. South Asia, particularly the Indian subcontinent, also showcases extraordinary genetic diversity due to numerous historical migrations and cultural interactions. The mixing of indigenous populations with groups arriving from the west and east has created a rich and complex genetic mosaic. The Americas represent another key region of genetic mixing, especially since the arrival of European, African, and Asian populations over the last few centuries. This has led to highly admixed populations where ancestral contributions from multiple continents are evident. East Africa, recognized as the cradle of humanity, also demonstrates significant genetic mixing due to ancient and more recent migrations across the continent. These areas vividly illustrate how repeated population mixing leads to a blending of genetic traits and widespread diversity. The concept of modern intelligence, as we typically perceive it, is primarily a result of cultural evolution. It is not necessarily due to recent biological changes in brain capacity or intrinsic genetic superiority. Instead, it reflects the cumulative advancements of human societies. Scientific consensus indicates that human brain size has remained relatively stable for approximately 200,000 years, since the emergence of anatomically modern Homo sapiens. This suggests that the dramatic cognitive leaps seen in recent history are not attributable to further biological enlargement. Several critical factors contribute to this perceived cognitive advancement over time. Advancements in education systems, from formal schooling to widespread literacy, have profoundly shaped human cognitive abilities. The structured transmission of knowledge enhances critical thinking and problem solving. The development of written language was a revolutionary leap, allowing for the externalization and preservation of complex information. This invention transformed humanity's capacity to accumulate and transmit knowledge across generations, fundamentally altering cognitive demands and capabilities. The continuous accumulation of knowledge over countless generations builds upon prior discoveries and innovations. Each generation inherits a vast repository of information, enabling further specialization and accelerated progress. This cumulative culture is unique to humans. Technology also plays a crucial role, amplifying human cognitive abilities. Tools, from simple levers to complex computers, extend our physical and mental reach, allowing us to process information, analyze data, and solve problems far beyond our individual biological capacities. Improved nutrition and health also contribute to enhanced cognitive function across populations. Better diets, sanitation, and medical care support optimal brain development and lifelong cognitive performance. These societal improvements have broad impacts. A Stone Age human, if raised in a modern environment with access to contemporary education, nutrition, and social stimuli, would perform comparably to a modern child. This underscores that cognitive potential is largely universal, with environmental and cultural factors shaping its expression.